I want to start with thanking the meeting organizers for putting together such a wonderful meeting. And it's truly an honor for me to have the opportunity to join the lightning presentation. As you can see from my presentation title, I'm here to speak to the application of CRISPR to the brain disease, in particular, the challenging neuropsychiatric disease research. As everyone knows, CRISPR-Cas9 system is a powerful gene editing tool, and it is supposed to hack out the disease genes in our DNA. However, during the past decades, as you can see from this slide, for many human common diseases, we have witnessed an explosive growth of genetic data by revealing even hundreds and thousands of genes that might associate with the diseases. Similarly, psychiatric disease also consists a long or huge list of genes. Besides this challenge, we have another bigger question: which cells in the brain we should target to? In other words, which cells in the brain are actually using the genes in question? Without this understanding and knowledge, it is very difficult for us to design a rational treatment for the diseases. As a neurobiologist, during this period, how we can overcome these challenges, because we cannot wait here until the cell or human cell adults, that type of projects to come up and give us really the complete image of the cell identities. So currently, we highlight the value of studying brain circuit function, because we all know our behaviors are controlled by brain circuit activities. Here, I'm going to highlight a brain circuit function, which we call thalamocortical circuit. In particular, I'm going to focus on a keynote called thalamic reticular nucleus, TRN, which strategically located between cortex and a subcortical region called thalamus. Why are we interested in studying these thalamocortical circuits? Because it is already found to play an important role in regulating sensory information processing, our sleep stability and architecture, as well as attention selection. We all know these type of symptoms are often seen across many psychiatric disorders. So we think that studying the brain circuit function could serve as an intermediate level to linking genes to behaviors, which potentially could give us the hope to work as a convergent pathways to regulate these potential defective brain circuit to eventually develop a novel therapeutic strategy. So how we can identify the TRN neurons' identity? So we apply a very powerful technology called single-cell RNA sequencing, which sort of give us a very clear idea about what the cells are, what type of genes they express. So for this purpose, we took advantage of an interesting mouse line, which labels specifically this small, thin, and irregular shaped TRN, as I mentioned, which is very important in this circuit. So by labeling these cells with red fluorescence, so we applied the fluorescent microscope and perform microdissection to scoop out this brain tissue, and then perform the single-cell RNA sequencing. After we know what are the genes expressed in these TRN cells, we perform computational analysis based upon how similar the genes expressed in different type of TRN neurons. Then we can interrogate these genes, indeed, TRN cells, we found that several disease genes identified from large-scale genetic studies are enriched. Besides, we also interrogate the genes which specifically expressed in some type of neurons, but not others, which potentially may give us an interesting molecular targets for the future regulation of TRN neuron function. Now it's time for us to apply this powerful CRISPR gene editing tools by designing the single RNA library to target these genes as we listed in this panel B. We know they are either disease genes or enriched expressed genes in some TRN neurons, but not others. And after make the AAV library packaging, we took advantage of this mouse line that already express this endonuclease enzyme, which cut the DNA strand. So then we just give an injection of this AAV library targeting to the genes we are interested in to study. And as an electrophysiologist, we then perform the electrophysiological recordings to see what the targeted genes contribute to the neuronal firing activity. For this purpose, we already know because these TRN neurons have this characteristic repetitive firing, which has been considered to play an important role in regulating the sensory processing. Specifically speaking, we already know if the TRN neuron firing within this physiological way, so they can suppress irrelevant informations, allowing you stay focused. For example, I'm talking, it means that your TRN neurons are firing properly. You can focus on my talking by suppressing the irrelevant information. But a lot of psychiatric disorders, patients cannot stay focused. This is just an example to show you the characteristic of firing, we call the repetitive firing signature of the TRN. That's what we start focusing on characterizing. When we look at these different pools of the AAV based upon the genes to catalog based with the similar expectation of the firing activities or synaptic transmission activities, we found that pool 3, when you knock out these genes, the TRN neurons show extremely active firing activities. Because, as I mentioned, in some of the disease animal models and even clinical patients, we already know the TRN firing activity, this repetitive bursting, is significantly reduced. So we are hoping that to start with the pool 3, it might give us a hope to regulate this firing activity. So then, when we look at the pool 3, give this significantly enhanced repetitive firing, we further design the single guidance RNA to target the individual genes to really pin down which gene actually is contributing significantly to this firing activity changes. Indeed, actually, we luckily found the KCDN2, which is a potassium channels that allow us to enhance the firing activity. This is just a strategy I want to demonstrate that by applying this CRISPR-Cas9 system, we are potentially not only identify the molecular mechanisms and potentially can give us therapeutic targets. I'm going to end with this acknowledgement. This is a quite a collaborative project, and being supervised by me and Joshua and Guoping, and the electrophysiology is mainly provided by Violetta, and a single RNA library and CRISPR knockout efficiency test were by another postdoctoral fellow in Qing Li. Thank you all for your attention.